Okay. Now, after noting that we have the oxidative phosphorylation, we also have the photophosphorylation. I want to leave an assignment where we compare oxidative phosphorylation with photophosphorylation. Compare oxidative phosphorylation with photophosphorylation. We've seen oxidative phosphorylation in respiration, photophosphorylation that is within autotrophic nutrition, particularly during the light stage of photosynthesis. All right, please make sure that you do that comparison. Remember, compare requires us to give similarities and differences. Similarities and differences. Okay. Now, looking at a summary of the whole process of respiration. Remember, we've said it occurs in 3 stages: glycolysis, Krebs cycle, and the electron transport system. That is, if it is aerobic. In other words, for a glucose molecule, when it enters the cell, particularly into the cytoplasm of a cell, glycolysis occurs, leading to formation of pyruvate or pyruvic acid. Pyruvate or pyruvic acid. Now, in absence of oxygen, when there is no oxygen, anaerobic respiration takes place, leading to a process called fermentation. Ethanol or lactic acid being formed. Ethanol in plants, lactic acid in animals. That is in absence of oxygen. But in presence of oxygen, under aerobic conditions, what happens to pyruvate? Pyruvate then undergoes changes that lead to the Krebs cycle. The decarboxylation, the dehydrogenation, forming acetyl-CoA, acetyl coenzyme. Remember the acetyl-CoA that we formed that is fed into the Krebs cycle. All these now occur within the mitochondrion, meaning the pyruvate is now fed into the matrix of the mitochondrion for the Krebs cycle to take place. After which, even the ETS will occur on the stalked granules, leading to oxidative phosphorylation, hence formation of energy or formation of ATP. Now, when we get a question asking us to describe the formation of ATP or the formation of energy from a glucose molecule, how energy is formed from a glucose molecule, or describe the process of respiration in presence of oxygen. How do we answer that question? Describe glycolysis in detail, just like we did. Describe Krebs cycle in detail, just like we have done. Now, when it comes to the ETS, don't give it in detail. Simply tell us that after Krebs cycle, the electron transport system takes place, where the NADH2 molecules in the ETS liberate 3 ATP molecules, and the FADH2 molecules liberate 2 ATP molecules. Meaning that question does not require you to go into the details of the ETS. When do we need to go into the details of the ETS? If the question is particular, that describe the electron transport system, then there we give it in detail, just like we have done. Or describe the formation of ATP in the ETS. Again, there we need it in detail. Describe the process of oxidative phosphorylation. That one requires the ETS in detail. So that is about the process of respiration, whether aerobic or whether anaerobic respiration. We now move to chemiosmosis, or what we call the chemismatic theory of ATP synthesis. We have seen the electron transport system, the ETS, and we said that when electrons are being transferred from one carrier to another, we saw that ATP is formed or liberated. We also noticed that when hydrogen atoms were transferred from the NADH2 to the FADH2, before going to the CoQ, there was also liberation of ATP. So how is that ATP synthesized during the electron transport system? That synthesis of ATP in the electron transport system is what we briefly call chemiosmosis, also called the chemismatic theory of ATP synthesis. How does it occur? Remember, we have seen the mitochondrion, and in the mitochondrion, after the Krebs cycle, that is where we have hydrogen atoms. But the hydrogen atoms are combined with NADH2 or FADH2. What happens to them? By the way, chemismatic theory was put forward by a scientist known as Michel, P. Michel. He's the one that came up with the chemismatic theory. So we are noting that hydrogen atoms are now combined with NAD and FAD in the matrix of the mitochondrion. This is at the end of the Krebs cycle. So in the matrix of the mitochondrion, the NADH2 splits and gives off the hydrogen atoms. In other words, we form NAD and hydrogen atoms. The hydrogen atoms then split into hydrogen ions and electrons, just like we're seeing it here. We said that the hydrogen atoms in the matrix after the Krebs cycle and glycolysis are within the matrix as NAD or combined with NAD in form of NADH2 or FADH2. The NADH2 splits into NAD and the hydrogen atoms that later split into hydrogen ions and electrons. Meaning at this stage in the matrix, we are having hydrogen ions or protons, then we are also having electrons in the matrix. However, these protons or the hydrogen ions are then pumped actively. They are moved from the matrix into the intermembranous space. Intermembranous space. Remember, we saw the structure of the mitochondrion, which had the inner membrane and the outer membrane. The inner membrane and the outer membrane. Now, so that space between the inner membrane and the outer membrane is what we called the intermembrane space. So when we look at this visual there, we can see the inner membrane, we see the outer membrane, we see that space in between known as the intermembrane space. Therefore, we are noting that the hydrogen atoms in the matrix split into hydrogen ions called protons and electrons. The protons are actively pumped. They move by active transport across the inner mitochondrial membrane into the intermembranous space. When we say they are actively pumped, or when we say that they move by active transport, it means that the process involves the use of energy. It involves the use of energy. So at this point, the protons have been actively pumped into the intermembranous space. Where they accumulate, they become too many because they are all pumped from the matrix, getting into the intermembranous space. So they accumulate in the intermembranous space. In other words, their concentration in the intermembranous space increases, becomes too high. This is why the pH in the intermembranous space is lower than that in the matrix. Why? Because the intermembranous space contains very many protons. That have accumulated there because they've been actively pumped. So at this point, we are having a high concentration of protons in the intermembranous space, just like we are seeing in our visual there. What follows after? Now, due to the high concentration of hydrogen ions or protons in the intermembranous space, it creates a great concentration gradient between the ions in the space and in the matrix. But at the same time, the inner mitochondrial membrane becomes impermeable to hydrogen ions. It becomes impermeable to hydrogen ions, meaning they cannot cross back to the matrix across the inner membrane because it has become impermeable. Instead, on the cristae of the mitochondrion, there are channels known as the chemismatic channels. They are channels called the chemismatic channels. Now, because the inner membrane is impermeable to the protons from the intermembranous space then move across the channels which we are calling the chemismatic channels. We have many protons in the intermembranous space, but the inner membrane of the mitochondrion has become impermeable to the protons. But there are channels known as chemismatic channels on the cristae. Therefore, the hydrogen ions of the protons then diffuse from the intermembranous space across the chemismatic channels back to the matrix. Why are we saying they diffuse? Because they are moving from the intermembranous space where their concentration is high to the matrix where their concentration is low, but across chemismatic channels. Now, in the process of movement across the chemismatic channels, there is synthesis of ATP. Remember, how is the ATP formed? ADP, adenosine diphosphate, combines with an inorganic phosphate. In other words, ADP is phosphorylated. There is addition of an inorganic phosphate forming ATP. But ATP is formed in the presence of an enzyme known as ATPase enzyme. ATPase enzyme. Very, very interesting. ATPase enzyme. Now, ATPase enzyme, where is it found? We say ATPase enzyme is found in the bulbous ends of the stalked granules on the cristae. The stalked granules are what we call the oxysomes. So they are bulbous ends. That is where we find ATPase enzyme, leading to formation of ATP. And at the end, the protons move from the membranous space to the matrix, where they recombine with the electrons that were left there. Remember, at the beginning, hydrogen atoms split into protons and electrons. Now, the electrons which were left there recombine with hydrogen ions, leading to formation of water. because the electrons combine with the hydrogen ions, forming hydrogen atoms, which then combine with oxygen, leading to formation of water. In summary, what do we mean when we talk of chemiosmosis? Hydrogen atoms in the matrix of the mitochondria split into hydrogen ions and electrons. The hydrogen atoms are from NAD